This is Audiomunity, a podcast about our body's never-ending fight with the outside world. Hello, this is Audiomunity, episode 48, recorded on November 4th, 2025. I'm Kevin Bonham, joined as always by the disembodied presence of Matt Woodruff.
Yeah, I'm going to haunt this podcast today.
You're a few days late for haunting, Matt.
I felt like I was in the spirit anyway.
Okay, fair enough. And then special guest today, we are joined by Zach Hilt, who's an assistant professor at University of Toledo. Hi, Zach.
Hi, thank you for inviting me today.
Yeah, absolutely. It's great to have you. We've been talking about this for a little while, that we want to try to get some other voices on the podcast, but I've sort of met you before. We have this online community that we're both part of, which is how we got in touch. But I would love for you to just tell us a little bit about yourself, who you are, your background, and what brought you to immunology.
Yeah, thanks for, again, having me on the podcast. And so I'm a very freshly, newly minted assistant professor. So I just started August 1st. So my lab is
Congratulations.
still, thank you. So still unpacking boxes with my lab manager. And, we have one, flask of cells growing. So we're slowly
big deal.
Nice.
getting things going. But I guess my little bit of my journey here is that I did my undergrad in just biology at a really small liberal arts school called St. John Fisher College in Rochester, New York. And so then I decided to move about 5 miles down the road to the University of Rochester at their medical center, where I did my PhD in a really unique PhD program called Translational Biomedical Sciences. And so it was a really interdisciplinary PhD program where people were doing public health, like clinical, bioinformatics, wearable devices. And
Cool.
I was like, yeah, it was really cool. It was really great training because on one hand, I would sit in a like super basic signal transduction class, and then in the next class, I was learning how to design clinical trials. And so it was a really great experience.
Awesome.
And then I joined the lab of Craig Morrell, and it was a great lab, great experience. So we have a great relationship still. And he was really interested in the immune function of platelets, which is something that most people don't think about. We think about platelets as, things, that are important for clotting, right? When you get a cut. And
I think
so
of them as cell debris, basically.
Yeah, they're just
Yeah.
death. They're death floating around.
we believe they're cells. And so my one pitch for the platelets is that they make proteins de novo. So I don't know how you define a cell, but there's no nucleus. And so we had a really fun project looking at monocyte macrophage interactions with platelets and how that regulated cardiovascular disease. And then actually how I found my postdoc, or what got me interested in being a hardcore immunologist. So really I was just doing immunology in a cardiovascular department, playing an immunologist. And so I was doing a side project with a clinician who was like a pediatric fellow, and she was really interested in what are the differences between neonatal and adult platelets, right? With the idea that we transfuse adult platelets into neonates, but they must presumably be different. And so we had a really cool side project on that, and a lot of that work is now being published by a postdoc in Craig's lab. But and then I was just like really struck by how different neonates were than adults, and it was like, they're just not tiny adults. And so serendipitously, I got forwarded a email from my postdoc advisor, Brian Rudd at Cornell University in the Ithaca campus, and he was studying, what are the differences between, neonatal and adult T cells. And so I said, that sounds really cool. And so then I, pivoted and became a hardcore immunologist, and so went down the T cell route. And so now within my own lab, I'm still interested in T cells mostly. I'm trying to integrate some of my myeloid background as well, but mostly my interest is kind of twofold. One, why are neonates, susceptible to certain infectious diseases? And so my number one interest is cytomegalovirus, or CMV, mostly just because it, causes congenital birth defects. but when an adult is infected, they're asymptomatic. And so kind of trying to understand why is it that, neonates are, uniquely susceptible to those types of infections. And then on the other hand, I'm also interested in like the long-term non-communicable disease aspect of being infected with a persistent latent infection. So, when CMV or other herpes viruses infect you, you harbor that infection for life, essentially, right? And so one of my, ongoing projects is trying to, re-implement that cardiovascular background with my immunology background, since it's kind of like a unique melding of the two, and trying to understand how these infections long-term have an, influence on cardiovascular disease.
Cool.
Very cool. Very cool. Did you happen to know Andy Varble while you were at U of R?
No, so we both went to the same high school.
Did you really?
Yeah, we did.
Okay.
But I've never overlapped with him ever.
Interesting.
Matt did his undergrad at RIT, so
Did.
Rochester
Oh,
Institute of
okay.
Technology.
And actually, I was I for a second was thinking about going to St. John Fisher,
Okay.
playing on the baseball team actually there, and then I ended over at RIT, and I was in Andy's undergraduate lab there, and then I know that he sort of left and then came back as a faculty member at U of R.
All right. Well, Rochester connection established, and now Matt can F off, and we'll talk about Tregs. So the paper that we're doing, so one of the ideas of inviting guests is to bring, Matt and I have our own little hobbies in terms of immunology concepts. We both went to graduate school at the same place, and so we were trained in a similar way. He went off and did innate immunity, or adaptive immunity. I went off and did innate immunity, and then now I do microbiome stuff. And so I've got my own perspective. Matt has his, and we're really interested in bringing on additional perspectives. One possibility is to promote your own work, but what you've done is you've brought a paper that is related to your work but is not your work, which is also very cool. So we get the perspective of an expert on a totally different subject. So this is going to be really helpful, I think. The paper we're doing is published in Science Advances. It's called Neonatal Regulatory T Cells Persist into Adulthood Across Multiple Tissues with High Enrichment in the Skin. And the first author is Morgane Hilaire, and the last author is Benoit Salmon, although it seems like both Morgane and Benoit are corresponding authors here. So I'm assuming, do you know these folks at all?
I don't.
No. Okay. So I'm assuming that the first author here did this work, started this work as a postdoc, and then started their own lab and sort of wrapped it up. So they're co-corresponding authors, first and last. I should say this was published last month, October 3rd, 2025. And this is like, you were talking about hardcore immunology. This is like, for me, I haven't thought about things like fate mapping and cell ontogeny for like 15 years. Although it was all the rage, I feel like, in the 80s and 90s, and has continued to have had its adherence. So this is the idea. Oh, go ahead, Matt.
No, I was saying I think that there was this critical moment, right? So the fate mapping stuff, tamoxifen-induced genes and all of that had this moment where it intersected with the brainbow mouse. And I think, Kevin, while you and I were in grad school, that sort of hit in force because, in the neuro side, brainbow had been increasingly used from an imaging perspective to, track neuronal development and lineages there. And then people realized that you could start using it for ontogeny studies in immunology. And so then it got really complex, and that's where Gabriel Victora's work, tracking individual B cells into germinal centers and looking at clonality and all of that stuff started to really explode. And Kevin, I don't know if you remember, but, in the late days of my PhD, it was all stromal cell development and, where are stromal populations in the lymph node developing from, where are the precursors. So it feels to me like, this is just kind of along the logical march here.
Yeah. Yeah, and I feel like those ontogeny studies really were about when you have so we know about like naive T cells, naive B cells, they get activated, they develop. a lot of the early ontogeny stuff was just like starting from stem cells and saying stem cell beyond. And so what this paper seems to be is, rather than thinking about the fate of individual cell types, like really specific, I want to know I'm going to get cell surface markers wrong because I don't think about these either, but like my CDXYZ positive, blah, 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 negative Treg cell, like where does that go at each step? Instead, this is like lifespan ontogeny. Like you said, we're seeing at what stage of neonatal development are these Tregs coming in. And so maybe first, Zach, maybe you can just describe the technology. We've talked about some of these like fancy mouse models and genetic systems in the past, including, I think, tamoxifen induction. We've definitely talked about using Cree to turn genes on and off. But I would love for you to just explain, like, what is the underlying technology here that we're starting from?
Yeah, and so I guess one of the reasons why I picked this paper because it's very similar to a paper I recently published, but looking at the CD8 T cell angle and with infection with cytomegalovirus. And so they're both based on like a Cree ERT2 mouse, right? And so how that works is that when you ERT2 is a tamoxifen receptor, right? And so it's an estrogen receptor, and so when you give tamoxifen, it activates that receptor, and so then activates the Cree recombinase. And so what we have done in the past in my postdoc biologist lab was that we had a TCR delta flocks, or promoter, and then so when we gave tamoxifen and it would fluorescently label all T cells undergoing recombination within the thymus. So then you could stably track them by flow cytometry.
And critically, this is not like labeling like with a dye. It's permanently turning on a gene. So you treat transiently with this drug, and then that gene is now on for the rest of that cell's and all of its descendants' life.
Yep. And then so like we were able to give tamoxifen to pregnant dams, and then or to lactating dams, and then they would transfer it to pups, and then you would be able to stably track T cells made at the time of birth all the way through adulthood, right? And so it was a really valuable tool to, one, know that T cells even persist from birth, and two, just know how they respond to infections. And so that's really a lot of what my postdoc advisor was doing, and then what I continued on as well. And so in this mouse model, they do something a little bit different where they're utilizing a fate mapping mouse model, but this time they're using a FOXP3 inducible Cree. And so they cross them with the Cree ERT2s. And so how this works is that then when you cross these mice and you give tamoxifen, the LOXP is flanked. It flanks the stop codon, and so then when you give the tamoxifen, the Cree will cut out that stop codon, and then now any cell that expresses FOXP3, so Tregs, since it's a master transcription regulator, will now be fluorescently marked indefinitely for the whole life of the cell. Even and the important part here is that when the T cell replicates, it still maintains this fluorescent signal, and so you can stably track these T cells over time.
One of the things that I don't think I realized, they mentioned it in the introduction, is that mice are born lymphopenic, right? So they have very few T cells.
Yeah.
So I'm gonna contest the word lymphopenic here.
Okay, but thank you. I agree. Okay, go ahead.
Hold on. I was getting there. Calm down. Just ran over my whole point, Matt. Constantly do. That's fine.
I had an immediate and an aggressive reaction to the first sentence of this paper.
Yeah, no, that's fair. That's why I wanted to bring it up. So I feel like I've lost my train of thought. Lymphopenia. Why is that wrong? Go.
Yeah. Well, go, Matt. I guess go ahead, Matt.
Yeah,
you sound like you had a point.
Yeah. So I'm gonna show my B cell colors here for a second, because a lot of good work has been done around this, right? And when we compare things to adult intact immune systems, right? and this happens in infection all the time, right? We compare sort of steady state to altered state and those sorts of things. And we use words like lymphopenic that inherently suggest that something is wrong with the system, or that the system is not yet functional in some way. And, just speaking from B-cell biology, and Zach, I'd love to hear your perspective from the T-cell side, because this is something that I know much less about. There are B-cells when these pups are born. They've already engrafted in peripheral tissue, and in fact those B cells have a very discrete function from the B cells that will eventually come later, right? So on the B cell side, we talk about the difference between B1 B cells and B2 B cells. We know that you can get B cells from we actually know that you can get full immune compartments that engraft into the skin, into the spleen, all these places from non-hematopoietic origins even, right? So you can get macrophages, for example, that do not come from HSCs, and in fact they develop completely independently of the way that we think about the development in a normal, healthy adult, right? But none of that means that the system is not functional. If you look at those B1 B cells, for example, that engraft in neonates, they have much more restricted B cell receptors, but those B cell receptors have a tendency to be very focused on historical pathogens that seem to be really important in the survival of the species, right? Mm-hmm. So to say that they're lymphopenic and in sort of assert that it's not developed is really incorrect. It's developed for that developmental stage of the mouse. And again, that's all B cell and innate perspective, but I'd love to get the T cell side of that story.
I agree with that. And so I think what people focus on is the number of T cells, right? And so if you pull out a day 0 or day 1 neonate, yeah, there's very few T cells, right? But the lymphopenic makes it sound like there's none, but I can mash up a neonatal spleen and get out enough, and if we're using like a transgenic mouse, right, I only need to put in like 200 T cells to get a response, right? And so I can get enough T cells to do a transgenic, TCR experiment, right? And so there are T cells, it's just very few. And so I guess the key difference...
So this is, once again, the tyranny of the FACS machine.
Okay, yeah. I think the
shaping
the major difference is that when a day 0 newborn mouse is born, their immune compartment looks like more like a late second trimester, early third trimester human, right? And so in humans, T cells start to appear at about 14 weeks of gestation. And so, T cells don't really seed, until they're born, essentially, right? And so those are like the biggest, differences between humans and mice, is that it's when a mouse is born and it's really still more developing like it would still be in utero.
So that was actually where my question was going, is comparing this to human development of the innate or of the adaptive immune system. Mm-hmm. And I think, Matt's point about this is an evolved response. Like, if it were important for neonates to have more well-developed immune systems, evolution could have figured that out, right? And there's a reason. There's clearly like some trade-off happening, and for mammals at least, we are getting passive immunity from mothers, and so I know that's true of B cells, of antibodies, for example. I know there is some cellularity in breast milk, but I suspect that T cells themselves, or at least active T cells, are typically not being transferred in quite the same way. And so, have people looked at, is there a T cell? Let me back up a sec. I'm going to cut my lead in to that, because I'm trying to formulate a question about the differences between T cell and B cell immunity in neonates. Matt, what am I trying to say?
I do not know.
I think I got it. Okay, so we know that in breast milk, antibodies are being transferred, so we're getting some passive immunity from moms. The same is obviously not true of T cells, at least not to any great extent. So in terms of the functions of T cells, here we're talking about Tregs, but obviously you've talked about cytotoxic T cells. for a neonate, whether human or mouse, which T cell activities need to come online first, and what are the implications there for, the health of neonates?
Right. And so, yeah, neonatal T cells are fundamentally different. And so I can describe a few of the ways that they're unique, but I guess fundamentally they're more like an innate cell than their adult adaptive counterparts, right? And so so a lot of that stems from, fetal and neonatal T cells arise from fetal liver HSC origins, right? And so versus your, adult T cells that arise from your bone marrow HSCs.
HSC, I should say, hematopoietic stem cell.
Yeah.
We've said that a couple times, just
make
sure people know what the acronyms are.
And so that was some of the work that, my postdoc advisor did before I ever joined was trying to fate map that. And he has ongoing interest in trying to understand non-hematopoietic origins as well, which is controversial in the T cell field. I'm not going there. But we do know that T cells do arise from fetal liver origins as well. And so that really drives a lot of their unique neonatal functions. And so we know from many experiments that neonates have increased proliferation. We know that in CD4s, they have a Th2-like skewing. We know that their TCRs are fundamentally different, and a lot of that's driven from a enzyme called TDT, which is what helps put in N additions during the VDJ recombination. And so because there is no TDT enzyme during neonatal period of life, it means that your T cell receptor, recognizes distinct epitopes versus adults. And so this has been really well described in CMV, right? So in the murine strain, neonates will recognize IE3 protein versus adults that will recognize more like IE1. That's in the minutia, but that's an example of, their TCRs are fundamentally different.
So, just to put a point on that. So TDT, you were saying, when the VDJ segments get rearranged, they're stuck together. That provides combinatorial diversity, but then TDT adds sort of random nucleotides into those joining places directly at the spot of the TCR or BCR where they're binding to epitopes. And so you're saying in the neonates, you don't have TDT, so you get combinatorial diversity, but not this additional diversity added by TDT.
Yep.
So you've got sort of whatever diversity you have that's possible is germline encoded almost.
Basically, yeah.
And do you also see, because this is what happens in B cells, same situation, right? But in addition to that, you also get V gene and J gene biasing. So what they see is that the usage actually of the repertoire is altered and restricted beyond there. It's not like an innate like lymphocyte. It's not that restricted, but there are clear biases, and be interesting to know if we saw that too.
Yeah,
I'm not familiar with that. That's interesting. Yeah.
Well, another sort of constraint here. I mean, we don't need to go into all of these details, but possibly interesting that for a B cell receptor, it's just about antigen binding. So evolution is selecting for VDJ segments that are going to bind what you were saying, Matt, historical antigen. But with T cell receptors, you have the added complication that they have to bind to their epitope in the context of MHC. So you need to have whatever your combinatorial diversity can see both peptides and also the MHC that they're binding to with a strength that's not so strong that they're going to attack self, right? That's it.
Yeah. And then I guess
maybe not interesting. I'm sorry.
We can just wrap up the neonates unique, but back to them being innate cells. I mean, they express TLRs, so we know neonatal T cells can express TLR2, TLR5. They have NK markers. The ways to activate them independent of their antigen. Essentially, they have complement receptors, and they even make unique cytokines. So in humans, they'll make IL-8. Mice don't have IL-8, but they can make things like IL-13, IL-11, just, cytokines that you don't think about when you think about a typical adult T cell response, essentially, right? And so, but in a lot of ways, it makes evolutionary sense, right? Because, it's less important to form memory early on in life, but really important to form effector responses. And so these neonatal T cells are really poised to form these robust effector responses at the cost of forming memory, essentially.
It's a cool perspective, yeah. All right, so so this starts with, you were describing before, you've got a cell where you can treat mice with tamoxifen, and then you permanently label cells that were alive at that time. I miss so FOXP3 gets turned on post like VDJ recombination, all of that rearrangement. So these are naive cells, but they're fully mature naive cells, right? They get induced, they're permanently labeled, and so now we can track essentially when these cells were labeled and look later. So maybe with that we can hop into figure 1.
Yeah. So for figure 1, they're utilizing their fate mapping mouse model. And so I guess even going to a supplemental, they do a really nice job in supplemental figure 1, just looking at their efficiency of the model and how long it works. I don't know if this was a reviewer comment. It feels like it, because like I don't ever want to do this type of work in fate mapping, but we get asked it, right? So, but they do a really nice job demonstrating that a lot of the marking is happening within the first 4 days after tamoxifen administration, and then by day 7 to 10, there's no like residual marking. So they know that once you give that tamoxifen at 1 week, after 10 days, we're not getting any more marking. And so it's a pretty good discrete window. And so they basically outline it as like almost like a week period when they set up their experiment. So A really shows that they have their 6 different groups, and so they're giving tamoxifen either at birth, 1 week, 2 week, 3 week, 4 week, 5 week, 6 week, and then they age the mice out into 6 weeks, or it's like 5 weeks, and then they age out to 6 weeks. And so then they're, looking at, how, what is the percentage of Tregs after they give tamoxifen. And so, they're showing that in all the different tissues, at least in B, that initially all of those Tregs are basically being marked. And so, their model is very efficient and very good. And so that's, a plus. And then I really like how they demonstrate their data in C. And I'm like jealous about their I love those like dot maps. So so I would encourage everybody to like look at the figures. And so 1C, they do this really beautiful like dot map where they it really helps you visualize the developmental layers essentially, right? And how
Although I want to say, I was reading this. I have this e-ink paper tablet, and that I usually do my reading on, and for some reason there's differences. Like I don't know if it's the transparency, but when you're looking at the colors on the e-ink tablet, they don't look similar, and so like the colors on the dot parts were different from the colors on the graph, and so I was like staring at this for like 5 minutes, being like, what are they trying to show here? I can't understand how these things are. And then I pulled it up on my phone and I was like, oh, okay, these are the same thing. So it took me more time than was reasonable to understand this figure. That's my fault, not theirs. But I just wanted to complain.
Yeah. And so what they're essentially plotting is that, by, after 6 weeks, is the, makeup. And so, the problem with these fate mapping mouse models is that you can only mark the mouse once and know when the T cell was made, right? And so that's one of the current limitations of the system, which makes a nightmare because then you have so many experimental groups. And so, but what they do with their 6 experimental groups is that they then take the percentage from that 1 week, from the 2 week, from the 3 week, pile it together at the end time point, and then say what percentage of the total 100% fraction is still marked from that 1 week, right? And so when they look out past 6 weeks, what they're finding is that those Tregs that were marked at 1 week and 2 weeks, 84% are still in the skin at greater than 6 weeks, post birth. And
where
so
84% of Tregs in the skin were marked at 1 and 2 weeks, right?
Correct.
Okay.
So bona fide neonatal Tregs, essentially, right, are persisting into young adulthood. And they do a nice job, looking at different tissues. I'm always interested in random tissues that people don't usually look at, but that's my own interest. But, these are, typical tissues that we don't really think about. But like in lymphoid tissues, this isn't as much the case, right? So like in the spleen, there's only 35%. So there's definitely some unique, tissue ontology that's happening within various organs, which is also quite interesting, right? And I think it's an open question in the field about what dictates difference in tissue immunity and tissue ontology. And
yeah,
go
one thing that I thought was interesting, the second most, let's call it neonatal organ that they looked at was the visceral adipose tissue. And I sort of think about, when I think about like sort of what's the right phrase here, tissue, I don't know, they're like, which tissues sort of go together in terms of trafficking receptors, in terms of function, pathogen exposure, all of that kind of thing. I sort of think about adipose tissue and colon and liver as being similar, but they have very, very different profiles here in terms of what percentage of Tregs are coming from neonatal. And there's obviously lots of other complexity in the immune response in these organs, but I just thought that was interesting that after skin, it's the VAT as being most neonatal.
Yeah, it makes me start thinking I should pull it. I've never pulled an adipose tissue in my life.
Let me quibble with interpretation though here in this data, because I actually think that there's something here that is important based on all that conversation that we just had. An interesting interpretation of this is, right, everything that they didn't fate map, they call greater than week 6. An alternative is that these things aren't being fate mapped because they're actually too late in the game. Right? So that first injection time point is week 1. If you look at the skin, it could well be that the remainder of those aren't labeled at any point during that because, not because, week 6 wasn't enough, but because they're actually too late to the game. And that was actually the really interesting piece of the liver, where we know a lot of this stuff develops, right? It could well be that the liver, actually a substantial portion of those cells are unlabeled because they were already there to begin with. And I think it would be really, really interesting to go back and ask the question, are these continuing to develop? Or, and I think it's actually a more reasonable interpretation of the data. If they're not seeing continued building at week 4, it could be that up to 30% of those liver-associated T cells were there when those mice were born, which would be fascinating.
Well,
that's a good point.
But in B, though, in the thing that argues against what you're saying, Matt, is that in B, what they're doing is they're tamoxifen labeling, and then they're pulling the organs 1 week later, and they're looking at what percentage are fate mapped. And so if you look there, in all of the organs, including liver, although maybe a little less at week 2, but when you pull just a week out, you've got like 90% of cells are labeled, right? And so if you're looking at the whatever it is, 31% unlabeled in the liver at week 8, when they were labeled at those other time points, you were getting 90% of those cells after a week.
So you're arguing they're turning over. So
they have to be, yeah.
Right. Interesting. Okay. All right.
Right, because otherwise in B, you would see that 31%. If they were pre week 1, you would see that 30% in B.
Right. It's not clear to me then how you would have that kind of turnover. So
because they're pulling at week 8. So
right.
even
so
at 6 weeks, so these are the ones that are developing between tamoxifen labeling at week 6 and pulling the organ at week 8.
Okay, so we're saying turnover is fast enough in the liver where week 7, 30% of those T cells are going to be developing and engrafting, and yet...
Well, we don't know about engrafting, right? Because if we looked at week 9 and 10...
Sure. Yeah.
It could keep sort of spreading out. One of the interpretations I had here is basically in terms of what's being fed by the circulatory system.
Yeah, we don't know what's tissue resident.
Right.
Correct.
So in the skin and in the VAT, I could imagine you're not having quite as much circulation happening from the systemic populations, whereas liver super infused with blood, same with spleen and lymph nodes. So Yeah.
So this one then would argue waves, right? So if everything you just said was true, and I believe that, I think I misinterpreted B a little bit, but it would mean that in the liver, for example, you have a stable population that is generated early in neonatal life that is functionally discrete from what's going to come later. So while, what comes later is going to turn over, right? It's going to be susceptible to sort of loss and turnover at that 30% level. You've got a discrete phenotype of cells that does seem to persist, which I now understand is consistent with their overall thesis.
Yeah, although in terms of persistence, we don't necessarily know persistence in the tissue, right? One thing about this figure that I was a little bit annoyed is that they don't look at blood. And I know Tregs probably are not a huge population in the blood, but of the Tregs in the blood, what percentage is coming? Because if you've got cells, we think about this often in terms of lymph nodes. You have cells that are coming in and leaving, and they're sort of surveilling. Same is probably true in spleen. And so I sort of think about lymph node and spleen, the sort of top two panels of C, as being essentially systemic Tregs. What do you have overall in the body? And so everything off of that line is the stuff that's potentially interesting. So colon looks effectively the same. Liver, you actually have a little bit more, maybe, of a drop off in terms of adding more after week 4 or so. But yeah.
Yeah, and that's
I feel
what
like we could stare at this for days.
We could,
all kinds of hypotheses.
Yeah, fair enough. We can move.
Yeah. Maybe we can move on to the next figure.
It's a good, it's a good figure when you just want to stare at it for a while, I guess.
Yeah.
Right. And so, basically their takeaway is that, there's some, interesting organs like the adipose tissue and, the skin where they seem to really persist. And so, from previously published literature, we know that in the colon, Tregs can express other transcription factors outside of FOXP3. And so we know from other studies that neonatal Tregs will express GATA3. And then we know that adult Tregs will express ROR gamma T. So they wanted to use their fate mapper to see if they could see if this held true in their fate mapping as well. And
these are the prototypical Th2 and Th17 transcription factors, respectively, right? GATA3,
yep.
Typically Th2.
Yep.
ROR gamma T, typically Th17.
Correct. Yep. And then so in A, they're just showing their two-way plot of ROR gamma T versus GATA3 or double negative population, and then looking across their populations and showing that their 1 week and 2 week marked adopt this GATA3 positive population preferentially, which makes a lot of sense based on what we know from the literature. And so kind of a nice confirmation. And so, while very few of them express only ROR gamma T, about 12%. And so then they in B want to compare that across, and so that was just the colon for A, since that's what published. And so then they wanted to look across all their tissues that they were polling and see if this held true for other tissues as well, or if this is really just a colon specific phenomenon. And so I think across all the tissues except the skin, interestingly enough, most of the early tagged Tregs had higher amounts of GATA3. Like in the adipose tissue, it's like almost 100% in some of the.
That's wild.
Yeah, and so which suggests that maybe right. One of the current problems, I get, the B1, B2 field has surface markers where they can distinguish them. In T cell field, we have no markers to distinguish neonatal and adult T cells. And it's a huge problem because every single study needs to be a fate mapping, essentially, right?
Sure.
Which is problematic for many reasons, right? These experiments are a nightmare to do, having done them myself. And so, if we can actually have markers that help you distinguish neonatal versus adult origin T cells, that would go a long way. And while that's not perfect, it does suggest that if you can identify GATA3 positive Tregs, a large proportion of them would be neonatal in origin.
Mm-hmm. Mm-hmm.
It's wild that the War gamete T phenotype is only in the colon. I mean, like, they're showing significance bars in some of the other tissues, but like it's effectively not.
Physiologically not relevant, right? But yeah, the War gamete is very it's interesting. I mean, I don't think about the colon a lot because I hate pulling it.
Yeah, no, that's fair.
No fun.
That's fair. It's it's the thing that I'm thinking about most these days because I study the gut microbiome, and so like I'm very interested in how gut immunity is then sort of like spreading throughout the rest. But obviously, there are some unique aspects to it. What is there a hypothesis about why? Like what these transcription factors are doing for these cells, because like I don't feel like we've gotten into transcription factors a lot on the podcast. Matt, am I misremembering that?
No, I think probably early on we talked about polarization and things like this, but I think it's been a while since we really touched on it aggressively.
But basically there are these sort of prototypical transcription factors that tend to label certain cell types. And this was all the rage identifying these, just before Matt and I got to grad school. But typically, the sort of like canonical thought is that if you have one of the transcription factors on, it suppresses the other ones because these are sort of driving the particular phenotype of cells. Honestly, like I don't actually know what the targets of most of these transcription factors are, but we said like FOXP3 is essentially like if you have FOXP3, you are a Treg. If you don't have FOXP3, you are not a Treg. I thought the same thing was true for War gamete and GATA3, but here clearly you've got some cellular programming for these T regulatory cells that is somewhat Th17-like or Th2-like. So is there thoughts about like what are the actual targets of the transcription factors that are like, and what behaviors these cells might have that's different?
I mean, so we do know there's some plasticity to this, right? So it's possible to reprogram a Th1 to a Th17 and vice versa, right? So there's definitely some plasticity to these. It's not just it's not like the textbook where you draw the arrow and it's definitive forever, right? And why Tregs would co-express more than one? In the discussion, I mean, to jump to the end, in the discussion, they hand wave a little bit that maybe, when the Tregs express GATA3 because of the Th2 polarization, right? It controls expression of a lot of these Th2-like responses. And so maybe when you have this suppression program, maybe it's a way to suppress Th2 specifically. I think that's what they kind of allude to in the discussion a little bit. I don't know if I totally buy that. And also like developmental origin wise, like in neonatal CD8s, for example, like GATA3 is upregulated in CD8s whenever I look at, early life expression, right? And so, but yeah, CDs are slightly different, but so there's also something developmental too, right? So maybe it's just because they come from a unique developmental origin and that they retain some of these, maybe epigenetic or something. I don't really know what fully controls that. It's beyond my expertise, but that's my hand waving.
Yeah, fair enough. All right, so we've got weird transcription factors in the gut. Again, this feels like a reviewer experiment because most of this paper is about the skin. This sort of like skin is the only thing that's not interesting in this figure, so it feels a little bit out of nowhere. But then, so figure 3, now they want to start looking at activity. Like, what are these cells actually doing?
Yeah, and so they're doing so they did a large spectral flow cytometry panel. So 25 colors, looking at a bunch of canonical markers that we think about that T cells will express. And
Matt,
so,
have we talked about spectral flow? I feel like you and I have, but I don't think we've talked about it on the podcast. This is like a newish fancy technology in the world of flow cytometry.
Yeah, I mean, we've talked about, I think, traditional flow cytometry a lot, and the idea that, fluorescent signals can be pulled away from each other just by looking at, where individual fluorescent proteins or molecules fluoresce, sort of capturing the peaks of fluorescence, and really just measuring fluorescent markers at those peaks and having to sort of subtract away all of the overlapping fluorescent signature. So I guess I would just lead with the idea that spectral flow cytometry is very, very similar. It's just that instead of trying to take the peak of where the fluorescent emission is and then subtract away everything else, what you're doing basically is you're analyzing the entire emission spectra of each fluorophore. Right, and so you're getting a much more complex picture of the emission across, however many channels you choose to do, and then there is a bunch of black box magic that happens behind the scenes that basically allows you to
Fourier transforms, Matt.
Yeah,
that's the
exactly. That is exactly what
transforms.
It is. Yeah. So it uses magic to then decouple or basically unmix.
Also known as math.
Those, yes, complex math that cannot then be reported on the back end of the spectral flow. And so, yeah, what you get is a much more greatly increased capacity of the number of fluorophores that you can basically use in your panel, because you're using much more complex math in order to pull those signals apart from each other. So
because you can have fluorophores that are effectively much closer together
functionally
on the spectrum and still be able to disentangle them.
Yeah,
right?
And I would say that is somewhat oversold. So yes, you can get a much higher density. There's no question about that. People still do make some pretty egregious mistakes where they believe that the overlap is not going to be substantial, and then they get some pretty serious problems. So, I keep using the word magic. The technology is not magic. You have to be really, really careful with controls in spectral flow, or else your unmixing goes haywire. And because the math is complex, people tend to miss the mistakes in the math. So anyway, it's a fancy advanced flow that allows you to get, like you said, Zach, 25 colors in a panel relatively easily. Not easily, but with a little bit of effort.
I mean, it just makes it possible, right?
Yeah, absolutely. Absolutely. Yep.
And so they generate a bunch of PCA plots, and so this kind of spills over also into supplemental. I actually kind of prefer the supplemental figure 4 a little bit, just because it did a nice job. So their markers both encapsulated conventional CD4s and also Tregs, and so then they use the PCA to see if they can do the organs and the type of CD4 T cell cluster together, right? And so their, PCAs and supplemental figure 4 do a nice job of demonstrating that, right? So, conventionals cluster together more versus Tregs, they cluster together more. And then the organs tend to cluster together. And then, more similar organs will also kind of cluster, right? And then I've never seen this technique done before, where they use the surface markers to generate the PCA plots and kind of show what surface markers dictate where the, different cell type and organs kind of what surface marker dictated how the PCA plot split. And so I thought that was, it's kind of hard to interpret by just looking at the PCA plot, but I thought it's cool you could do it.
Yeah, this is called a biplot, which is a weird name for it. But this idea of putting on a PCA sort of the vectors of individual components and how much they go along either axis. The length of the vector is the sort of force of the vector. It's the analogies to physics are a little bit strained, but basically, so you see Iacocque jumps out at me there. It's sort of up and to the right. So along the first two dimensions, Iacocque is sort of equally split roughly between those two dimensions. And PCA, I feel like we've talked about this before, but PCA is some weird stuff. I don't think it's worth going into, but effectively it's just showing how much these populations are separated by along multiple dimensions. So you're sort of collapsing multiple dimensions of data down into 2 dimensions, again, using some math. Or magic, as Matt likes to say.
Yeah.
Well, no, and I think Zach's point was that it's rare that you see PCAs on flow cytometry surface markers.
It's rare that Matt cuts out when he's trying to make an important
It's common,
point.
I think. It's common to see them in single-cell transcriptomics, right? They show up. Dimensionality reduction shows up all over the place in transcriptomics. I think it's rare that the PCAs show up on surface markers for a spectral flow panel. Although I like their style because we routinely used high dimensionality visualizations in some of the flow stuff, because I feel like when you get up to like 20, 25 markers like this, it starts to become really important to reduce dimensionality.
Mm-hmm. For sure. And so then in B, they pull out like specific markers that we think about a lot for T cell surface expression. And so in B, they're looking at CD44, which is, a canonical activation marker. I'm not going to say like antigen experience, or again, these are all naive mice, so they're uninfected. But CD44 marker of activation, CD62L, which is a selectin important for trafficking between lymphatics and blood. CD69, which is also a broad activation marker, or also a marker of tissue residency. Kind of depends. The problem with these markers is that they can mean different things in different contexts, right? And so it's always careful how you interpret these. And then BCL2, which is a transcript that helps with cell survival, essentially. And so go ahead.
No, I was just going to say, and what they're specifically doing is they're comparing the percentage of cells in these populations that are derived from the first week versus all of the other weeks, right?
Yep. And so we know these are bona fide neonatal Tregs, and in a way, comparing the bulk is kind of nice, because then it gives you what that this is very specific then, right? And so that there's not, and so is it like a progressive type of thing, right? But this is like very discrete. We know that these bona fide neonates are unique in this way. And so overall, they see that CD44 is higher, which also corresponds a lot with the conventional T cell responses. So like in the CD8 field, neonatal T cells will express CD44. And we often call these virtual memory T cells. And so these are T cells that
virtual memory.
I think Ross Kettle coined the term, which I think he might regret now. But essentially, it's a term where they look like memory T cells, but they've never seen antigen, and they actually behave a lot. Then they actually behave and they have they have more enhanced like effector functions, right? They respond faster to infections. So there's something unique about them. Here, they don't really show that, these CD44 positive ones are necessarily unique in function, but it's interesting that this holds true in Tregs as well. So there's something, again, back to probably developmental origin, where they're biased towards this. And then in the untagged population, they have more CD62L versus the neonatal ones, where it expresses very low levels, actually, which could explain why they probably go away over time too, and maybe they don't recirculate, right? They don't actually go back into lymphatics. And this also holds true with conventional T cells from early life, where they terminally differentiate more into like the end stage, where they'll downregulate 62L, will never upregulate it, because this is CD62L is one of those ones where up to a certain point you can up and down regulate transiently, but if there's usually a point of no return, essentially. I'm not sure how that works for Tregs exactly, but for conventional T cells, so maybe that suggests why these early life Tregs don't expand in other tissues, just because they're not going back into lymphatics and expanding, which is interesting. And then they're showing that also CD69 is slightly different, maybe, I don't know, 5% difference. And then BCL2 is also quite different, where the neonatal ones express a lot less BCL2, so maybe they're also not surviving as much. But in the skin, I don't think you can actually detect any expression, so I'm not sure if that actually is a regulator, right?
Yeah.
And then they're looking at some of these more, markers of activation, or, people think of these as exhaustion markers, essentially, right? But really they're just activation markers, and they only cause exhaustion when they're being stimulated chronically, right?
Right. Famous because they're targets for cancer immunotherapy, I think.
Yeah. Right. Yeah. So I like to call them markers of activation. I don't like the term exhaustion markers because you need to actually have functional. My pedestal is that you need functional data to call them exhaustion.
How dare
Yeah,
you?
Seconded.
How dare? Yeah, I know. How dare I?
I really believe.
Am I gonna get
in the
canceled?
existence-
Maybe cut this out. I'm just kidding.
No,
No.
I refuse. So, I mean, is there, like, honestly, so some people, when they stare at math equations, their eyes start to glaze over. Other people, yours truly included, start to glaze over when they're looking at lots of data from FACS panels.
Yes.
Basically, all of Matt's papers induce this phenotype in me.
Yeah.
So, in terms of the figure 3 take home, Tregs generate, like, what they say the take home is Tregs generated during the first week of life that persist into adulthood exhibit an activated phenotype. Does that sound right to you?
Yeah, and I think that's consistent with other types of T cells from early life. And so I guess to me the open question is what does that mean, right? And so as far as I mean, we can go into this later a little bit later too, but there's no suppression assays in this study, right?
Yeah.
And so is there something unique about their suppression function? Because we know CD44 positive early life T cells that are conventional behave very differently.
So the point is that Tregs function is typically thought of as being suppression, and what you're saying is we have no idea if these are actually suppressive T regulatory cells.
Yeah,
that's interesting.
So it opens some interesting questions, but
yeah, for sure. I guess. Yeah, I'm cognizant of the fact that we're coming up on the 1 hour mark here, and we have a lot more FACS panels to go through, which is a little bit troubling to me. I need to stop saying quite so much. I'm trying to notice. So they haven't activated. One of the questions I had about this activated phenotype, you sort of said offhand earlier that they haven't experienced antigen. My question is, do we know that that's true? Because we think about specific pathogen-free facilities as not having specific pathogens, but there are microbes. These aren't germ-free.
You're right.
Right? So we have, not only do we have microbes in the gut, certainly exposed on the skin, we also have potentially self-reactive cells that can be produced, even if they're not going to drive autoimmunity at some point. You still have potentially self-reactive cells popping up even at this early stage. So do we actually know that these things aren't antigen experienced? Particularly given that these, as we've said, are sort of more germline encoded. So presumably, evolutionarily, you might have a population of Tregs that's on the lookout for more self-reactive cells that are likely to pop up in early life.
No, 100%. Yeah, like you said, these aren't germ-free, so there definitely are pathogens, right? And so, like, this is tangential, but we've done some, like, dirty pet shop mouse models before, right? And so, like, when we submitted our clean, dirty, and pet shop mice for, like, bulk sequencing of, like, what they harbor, right? Like, you can find Staph aureus on our SPF mice, right? They're clearly not, pathogen-free, per se, right? And so, I mean, maybe that's along the line of the skin, right? The skin is colonized with bacteria, and so maybe that's why you're getting a Treg bias even in this study, right? Because there are microbes there. And also, I've gotten this comment too, where looking at antigen specific population than CD8 cells, and so like during our CMV studies, we'll have more antigen specific cells than could be accounted for with CMV. And so reviewers are often like, well, they're clean, they're clean mice. How do you have other antigen specific cells? It's like, well, they're not. It's not like there's no pathogens. I'd probably sneeze in the cage. I don't know. I'm just kidding. But so like, yeah, you definitely form a small amount of antigen experience for sure.
I mean, we know for a fact, right, just to simplify this, we know for a fact that if you take a mouse and you put it in a germ-free facility, it does not develop normally, right? And so, you obviously require that kind of contact in order to develop these systems. And then we pretend like that's not true when we're looking for things like antigen specific, right? Like we know that contact with those pathogens, or not pathogens, commensals at that point, right, must have had an impact and must be read out in these systems at some level. And then we get surprised when it is.
Right. So for conventional T cells, there is a marker CD49D, which is a very good surface marker for antigen experience. And so you can gate on double positive with CD44 and CD49D, and that's how we distinguish antigen experience versus activated not antigen experience. I'm not sure if that holds true in Tregs, but that could have been something, to look at.
And they didn't look at that here, as far as we know. So, all right. So figure 4, I feel like we can maybe blast through this a little bit. This is showing tissue-specific markers, most of which, honestly, I didn't recognize at all. I want to make 2 sort of data display comments on this figure, one of which is for the PCAs. PCA dimensions do not have dimensionality. PCA axes don't have dimensionality. There is clearly the 2 PCAs they show, the VAT and the skin, have very, very similar signatures, but the x-axis is reversed. Because when it comes out, the math, it just sort of, there is a reason why some values are negative, but you can just multiply everything by -1 and it doesn't make a difference in terms of the math. So I think they really should have flipped that axis so you can see the ways that they're similar. That's a very minor thing. The other thing I just want to notice, because it jumped out at me, is that in panel E, they're doing these essentially feature set enrichment kinds of analyses. And I just really like that the titles of the plots clearly were completely unedited because they still have underscores, and they clearly didn't clean it up. It just got spit out by whatever software they're using, and they're like, sure, fine, we'll just stick it in as is. I thought that was funny. So beyond that, I think we can skip figure 4.
Yeah. My only comment for that was that the fact that they retain expression of KLRG1 is somewhat interesting. It's a NK, like marker.
Oh, okay.
It's also expressed higher in terminally differentiated conventional T cells, and also early life T cells express it more as well. So perhaps KLRG1 expression could also be a useful marker for early life Tregs. So that's my only, takeaway from that as well.
Yeah, cool. All right, so figure 5. Oh, and we've lost Matt, but that's fine. So what's going on in figure 5?
Yeah, so when I first read this paper, I was going insane. I'm like, oh my God, they only looked at 8 weeks. It's not old enough. They're barely adults. And so then they finally age mice out to 28 weeks and go, how stable are these neonatal populations, right? And so, that's been one of the, questions in the conventional field, right? It's like in an uninfected mouse, these neonatal fetal derived T cells become less than 5% of the population by this age, right? And so are they actually important, right? And so they actually do a good job of, aging these mice out and confirming that, whether or not these persist. And so, the nice thing here, so they look in all the organs over time. And look at how stable they are. And so the skin, while it does decrease, but by 28 weeks, it's still about 40% of the Tregs from this first week of life are retained. And so it's a pretty significant, 40% is a lot, right? And so, that's convincing that these probably do matter. And then I'm also wondering too, these are all SPF mice, but what if they did dirty pet shop mouse experiments? Would we also bias the early life Tregs expanding also? And
can you say, sorry to interrupt, but can you say, you're saying pet shop? I don't think I've heard
pet
that.
Shop. Oh, yeah. I should explain.
Pet shop.
Yeah, pet shop. Yeah. So this was a model by Dave Masopos in Minnesota. And so they literally took mice from a pet store and they, so what they did was they co-housed them with SPF mice. And so then you would reconstitute their whole microbiome and everything in pathogens and whatnot from normal environment. And so essentially what they showed was that you now get tissue resident T cells at normal levels. And also their T cell compartment is a ton more, very small amount of naive T cells and a lot of effectors and antigen experience cells. And so Brian's group that I did my postdoc in, he's utilized that model also, and has demonstrated that if you expose mice early on in life, you expand these neonatal T cells, which is unsurprising,
Cool.
right? And so I think it also, the SPF conditions downplays the importance of this information, right? I bet these neonatal Tregs expand more in an actual dirty environment as well.
Mm-hmm.
Yeah, so that's cool. So you're taking advantage of the sort of microbial environment of pet shop, but then you bring it to genetically identical experimental animals so that you can sort of subtract out whatever variation is coming from genetic background, but still get all of that exposure. We did a paper a few months ago where they did wilded mice, so they had this like outdoor enclosure where they just like sent their mice out into the wild, quote wild, and looked at a bunch of stuff too. So that sort of seems like a
very similar model. I don't want to trap mice, so
that looks like a pain, that study.
Even the pet shop one is a pain. So with my PSA. But yeah, so they do a nice job demonstrating that these two do persist and that it's, somewhat organ specific. So the adipose tissue does decrease significantly, but the skin persists.
Yeah. Cool. All right. And then figure 6, now they're sort of zooming in specifically on the skin and talking about persistence and residency of these cells. So what's going on here?
Yeah, and so they use Ki67 as a surrogate for proliferation, although they do actually in the supplemental do EDU staining, and which is so there's multiple ways to stain for cell proliferation, right? Ki67 is an easier way just because you can do intracellular staining, and then but it just tells you if the cell is in the cell cycle. It doesn't actually tell you that it's divided. And then they do EDU staining in figure supplemental S7, where you give the T cells a dye, and if they if the dye signal decreases, then they're actually proliferating.
Because it gets diluted every cell division, it goes down by half, right?
And actually, for how EDU is, I think EDU actually gets incorporated, and so there's different ways. And so that was like cell trace violet gets diluted, EDU gets incorporated into the cell as it divides, and so then they actually divided. And so here they're just showing that, over time, the Tregs, initially the Tregs from early life are proliferating a ton. And then, it levels off over time, which is unsurprising. And we also know that early life T cells proliferate a ton. Like they're just programmed to completely, totally proliferate. We also know that in the conventional field, the overactive proliferation is one of the mechanisms by which they adopt this like CD44 high virtual memory like phenotype, right? And so that's probably all tied together somehow there. And so over time they proliferate less, not surprising, but probably just enough to maintain the population over time. And then here they're looking at in B, they're it's so currently in all the models it's unknown whether Tregs colonize the skin. Like when the Tregs are colonizing the skin, right? And so this is when they do the topical treatment, I believe, right? And so what they're doing here is they're taking HO tamoxifen and they're applying it topically to the skin of mice at 3 weeks to mark presumably Tregs that are within the skin. And so it should be able to, go through the skin and mark cells. And so here they're, just showing that from 4 weeks and 8 weeks when they're marking, the lymph node, decreases over time, but the skin's pretty stable. And so let's see. My only question was why they did 3 weeks for giving the skin tamoxifen. I don't know why they didn't give it earlier.
Is there something about the newborn pups like not having hair and their skin being much more sensitive? I suspect I wonder if there's an IRB kind of limitation.
Yeah.
You can do almost anything to mice, but I wonder if the tamoxifen painting on the skin for super early pups is just not gonna.
Yeah, that's what felt like to me. That feels like an Iacocque that wouldn't let them paint the pups before they weaned them out into their own cage.
Yeah.
It's just surprising because like we can like toe tattoo mice, like neonate mice,
Sure.
right? Because like you can't mark their ears right, and so
Well, it could be. I wonder if it's a technical thing too. Like maybe the skin. I don't know. Like estrogen might thin the skin and cause them to.
That's possible.
I don't know. It's interesting.
It is.
I did find it quite interesting in C, where again they do one of these PCAs on the cell surface markers, comparing skin versus lymph node at a couple of different ages. The neonatal Tregs in the lymph node are just drastically different.
Yeah.
But all of the Tregs from the skin are all sort of clustered. I mean, I guess that makes sense because we're saying that they are mostly derived from neonatal Tregs, but just it's a quite dramatic difference.
Yeah, I think it goes a lot on the lines. So Donna Farber at Columbia has done a lot of human tissue resident T cell studies, right? And one of her recent papers was really demonstrating that like tissue specific signatures in T cells is maintained over the lifespan, right? So there's like a core signature within each tissue that it just seems to be maintained. And how that is, who knows, probably just unique set of cytokines and microbial exposures, right? And so I think that kind of goes along with a lot of her human studies as well, where clearly, the tissue has a set program and you don't deviate, or you shouldn't deviate. Maybe that's why you get disease, right? If you deviate too much. So yeah, that's kind of what was my takeaway from that in the context of other literature, essentially.
Yeah. Cool. So take home here, neonatal Tregs are kind of cool. They're mostly in the skin by adulthood. I guess the VAT signature kind of goes away.
It does. I guess one of my takeaways too is that like in the paper they're like, oh, like they all go away, but really like in every other tissue it's like 5 to 10% are still there, so they're not like gone, right? And so there's still a population there. And we've even shown in the conventional T cells where even if it's a small population, they can still play a functional physiological role in the context of like infections, right? So whether or not they play a functional role still when maintaining at that level is an open question.
Yeah.
I mean, and this is, one of the interesting components of that field. I'm going to go back to the innate and the B cell side is, those cells frequently, they self-renew, right? So there's frequently a small stem-like population that kind of continues to allow those populations to persist. And one of the really interesting components of that is if you ablate those cells, you actually fundamentally change the way that the tissue responds. So frequently from a tolerogenic sort of predisposition to a more inflammatory predisposition. What's interesting about that is if you deplete like that, they do not come back, right? So if you deplete tissue-resident macrophages that engraft through that neonatal period, you do get macrophages again, right? It's not like the HSCs that are circulating can't reseed that tissue and turn into macrophages. It's that those macrophages that you turn into are phenotypically distinct from the macrophages that were there before, and that ultimately leads to differences in downstream responses. And that they don't explicitly do that here, but it felt to me like that's where we were headed, right? There's this resident population of regulatory cells that kind of persists. It looks like they might self-renew, and probably are physiologically distinct from the cells that come after. And I think teasing that apart would continue to be interesting.
100%.
So that's interesting, and I think you were saying one of the major limitations of this paper is the lack of functional assays for these cells. And a limitation in the field is the lack of cell surface markers for these cells. So what other ways can you sort of get at function? So I'm imagining you could have, instead of doing the labeling here, you could do diphtheria toxin kinds of things. So you can do depletions.
Yeah.
Obviously that has limitations as well. you can do adoptive transfer, maybe, but can you get these cells out, and then would they actually get back to the tissue if you try to transfer them?
Yeah,
Or
that's just a good question. I mean, you could. I mean, I don't know how many you need to put. The problem is always when you're not gaming the system with transgenic TCRs, you need to put a ton in,
Hmm.
and so it becomes very difficult to do those types of experiments.
Hmm.
And Tregs are a small subset of CD4s, right? And so it does become a challenge in how to actually model this. I'm sure. I guess my one thought is that we know that developmental lineage is regulated by like the Lin28B, Let7 microRNA pathway type thing. I'm assuming Tregs are probably also controlled by this, and so maybe you could game the system by doing one of those inducible mouse models. It's not perfect, but you can make them look like neonatal T cells.
Hmm.
We've done that before in conventional, so maybe that's a way to game the system. But yeah, and that's actually one of my ongoing questions of if you actually take out a tissue resident cell and adoptively transfer, does it go back to the same tissue? I don't know
Right.
if anyone's actually done that. Just I don't know if anyone's doing
Yeah,
that.
I imagine it would be
It's
pretty
so
hard to
fundamental.
get cell numbers
Yeah.
high enough,
Yes.
and presumably, trafficking from blood into tissue requires a different set of chemokine receptors and integrins and all of that kind of stuff than residing in a tissue.
Yeah.
A lot of the trafficking stuff was done in circulating cells that have to turn on and off. They're trafficking to get into the lymph node, hang out in the lymph node, and then leave and go to another lymph node. But I feel like tissue resident cells are probably they adopt the signature and then they're just there. They don't need to worry about coming back. So
Yeah. Yeah, that's one of the, yeah, I mean, like even I've done studies of like CD trafficking into the brain, and like, like I had a whole project on them expressing CCR9, and so, but what I noticed is that once they get to the brain, a lot of them down-regulate it. So would they actually ever get back to the brain? Probably not, right?
Right. Yeah.
Um,
Cool. Well, this was a cool paper. This is not one that I would have ever picked. So I really appreciate you coming on. Thank you very much, Matt. Did you have anything else that you wanted to follow up on?
No, I think it's really cool work. I'm excited to see where your work goes around CMV. I think that there's a lot of interesting avenues around how all of this eventually downstream impacts the ability of these systems to respond. And I think actually the intersection between that sort of handoff from fetal to more adult development and how those systems integrate, right? It's not binary, it is overlapping and parallel. And I think understanding those two arms is really fascinating. So I really appreciate you coming on and the opportunity to talk about all that.
yeah, thank you.
Yeah,
I'm always happy to spread the good word.
Once you have papers out from your lab, absolutely, you are welcome to come back on and talk about them, or your trainees. We would love to get postdocs and grad students on as well. So with that, this has been Audiomunity, episode 48. I'm Kevin Bonham with Matt Woodruff and special guest Zachary Hilt. If you like what we're doing here, please support us. The best way you can support us is by sharing us with your friends, other graduate students, other postdocs. Invite someone to come on. Tell them to send us an email. You can contact us at contact@audiomunity.org, AUDIOMMUNITY.org. You can throw us a few bucks per episode or per month at patreon.com/audiomunity, our website with all of our past episodes, links to subscribe, RSS feeds, etc. at audiomunity.org. And the music at the beginning and the end was composed by Rachel Reinick, and we'll see you next time.
Take care, y'all.
Sometimes you get lucky.